So OligoFit was really born out of the idea of trying to understand better the types of alpha-synuclein aggregates which exist in the different diseases. And it was, because we tried to make a vaccine to target alpha-synuclein. And when I asked the question, what is the target? They told me, oh, it's oligomeric alpha-synuclein. Subsequently, that term is too heterogeneous. And we now know that there's like maybe a great diversity of that species. So the idea was to see whether we could identify different shapes of these aggregates in the early form, so the pre-fibrillar form. And then if we could do that, whether we could then generate antibodies that would be able to discriminate. And in so doing, we would use the tools which we use to generate the antibodies to then create a therapeutic vaccine that targets that specific type. Brought together a really multidisciplinary team that was, to bring things like biosensors into the space. Biosensors are useful because they are rapid point-of-care tools, potentially. We also realized that we wanted to study the earliest parts of the aggregation reaction. And so we were able to partner up with some great people in Warsaw. And so they brought their photonics expertise to the group. And like that, we were able to see very early events in the aggregation cascade. So that was pretty cool. And then partnering that with some really established people in the field. So out of Göttingen and out of Denmark, in Aarhus, they basically brought this sort of the disease speciality and the assays and the cell models that allowed us to sort of be able to integrate and interrogate the toxicity or the misbehavior of these aggregates. And I guess the big successes was bringing 3 groups that were not necessarily neuroscience based into the neurosciences field, teaming them up with some great expertise in the field, getting everyone to this level where we could delve into what is a very rich and deep literature to ask these very pointed questions that are specifically looking at oligomers. I guess one of the other aspects was also introducing us to the world of synucleinopathies, people living with the disease. And JPND has a great sort of mantra of please include PPI in your presentation now. JPND was critical to this because I think I had never heard of JPND, and it was actually, not surprising, but I'm a vaccinologist. Vaccines for neurodegenerative diseases, there's a handful of them out there. We were actually contacted by our friends in Latvia who sort of made us aware. And when we looked at this program and we looked at what we could contribute, we very quickly understood what was it JPND was looking for. They're interested in sort of fostering a network of groups that are bigger than the sum of their parts. And I think that's what we were able to achieve. We brought new approaches, perspectives to other groups where they would never use this technology. They would never use this approach. And like that, they were able to test their models, test their approaches. It was, I think, generally positive. The other thing I was going to say about JPND is that it's obviously introduces you to a larger community as well. And the diversity of projects that they fund is actually very inspirational.